Okay, guys, we're back. I want to kind of start on the second portion of this wonderful trip through signaling. And we're going to now continue our lectures on signaling. And we're going to discuss, you remember those receptors we talked with our good Rastafarian friend? Okay, we talked about catalytic receptors. So we will discuss a lot of stuff here, and we discuss catalytic receptors. There are different types of receptors compared to the heterotrimeric protein receptors. Why? They're actually used in signaling by growth factors, numerous hormones, cytokines. They are the ones that will attach to these receptors. Catalytic receptors are single chain. So one single chain transmembrane proteins, so they span across the membrane. And what's going to happen? They will associate with another single chain or more, single chain usually with another, with two, single-chain transmembrane proteins upon the binding of the ligand. And these complexes will signal now, you remember, through phosphorylation of tyrosine residues on their tails inside the cell. These tyrosine residues get phosphorylated, and they will light up, send the signal through the cell. So phosphorylation of tyrosine residues, which we haven't mentioned, is actually achieved by a very important enzyme known as enzyme thyroid kinase. Now, why is this enzyme important? Some receptors will have their own enzyme. They will have thyroid kinase incorporated in them, while others, receptors will have to recruit another enzyme, thyroid kinase, from the cell and to do the job for it and phosphorylate the tyrosine residues. So they can have the tyrosine kinase, or they need to recruit the tyrosine kinase, and I'll explain to you why is that important. So this is how catalytic receptor signaling takes place. Ligand will bind to extracellular portions of the receptor protein. Remember that receptor protein is going to become phosphorylated on tyrosine residues. As phosphorylated tyrosine residues will now induce binding of other proteins. And these proteins, if you remember, will serve as adapters. They will bind to phosphorylated tyrosine and to another signaling molecule, adapter protein. And they will relay the signal deeper into the cell. Now, there is a problem, and it's not a problem. We know that every single signal doesn't need to last long. It has to be very brief. So the moment that there is a phosphorylation of tyrosine residues on the receptor, there's going to be dephosphorylation rapidly after signaling. And so this is achieved by protein tyrosine phosphatases, which will dephosphorylate phosphotyrosine. Oh my God, it's just an awesome statement, you know? So it's really, really awesome. This is going to be a question on your exam, exact statement. I'm just joking. And you're going to have to say it fast 4 times. But that's what it is. So that's why there is a very short-lived appearance of phosphotyrosines, and this is very brief signal that goes into the cell. So here are those receptors, okay? We see them this. And I'm going to show you now receptors that have their own tyrosine kinase. So these are the ones that have tyrosine kinase in them that they will activate. So we see that the receptors with intrinsic tyrosine kinase activity are used by numerous factors. Very importantly, they are used by TGF, transforming growth factor, epidermal growth factor, and platelet-derived growth factor, which you all hear in immunology extensively. For me, more important, and I think, is insulin signaling that actually occurs through these pathways. Now, receptors with intrinsic tyrosine kinase activity, what they're going to have? They will have external portion, which will attach to ligand, but then they will contain inside the latent or inactive catalytic enzymatic domain. This is the tyrosine kinase. And this tyrosine kinase will be activated when the receptor binds to the ligand. So tyrosine kinase is sitting waiting for a receptor to be activated, so it will turn on and phosphorylate tyrosine and will make those receptors pop out. This phosphorylated tyrosine will now activate the whole signaling process. So how does this happen is that most catalytic receptors are formed by association of 2 or more these single transmembrane protein chains. Usually it's 2. 2 single transmembrane chains will bind to ligand and to each other. That's the process of activation. And again, each of these single transmembrane chains will contain 3 domains. will have ligand binding portion when it binds to the ligand outside, hormone, signaling molecule, insulin. Then you have helical domain that spans across the membrane. And then you have effector region, which will extend into the cytoplasm. And this is the area where tyrosine kinase will be. They're intrinsic tyrosine kinase that will phosphorylate tyrosine on their end. So let's talk about catalytic receptor signaling, and let's go through the steps. I know, does it starting to make sense? We talked about it previously, so it's very, very similar. This is it. Ligand will bind to the receptors. How does it do it? It will bind to one chain and another chain. So it's going to kind of bind two chains together. Okay? So that is going to be dimerization of the catalytic receptor. So the ligand binding will bring these individual transmembrane protein chains together. It's going to bind for two. It's going to bring them together. Now what's going to happen? Tyrosine kinase on one will activate tyrosine kinase on the other. So they will cross-activate. Okay? So tyrosine kinase on one will activate tyrosine kinase on the other, and other will activate on the first one. So they become activated. And what happens now is now these activated tyrosine kinase will cause phosphorylation of tyrosine in their tails, both tails. It's kind of straightforward. So that is the first step of the signaling process. Next is this, is now we have phosphorylated tyrosine is now present inside the cells and on the tails of these receptors. Now what do they need to do? They need to bind to adapter or adapter proteins, okay? So these adapter protein will adapt, they will bind on phosphorylated tyrosine and on a secondary messenger molecule, okay? So they will be the one who will form that bond. So this is it. And why they can bind to this phosphorylated tyrosine? Because they have SH2 domain. SH2 domain on these adapter proteins will enable them to bind to phosphorylated tyrosine. Okay? Now these adapters attaching to phosphorylated tyrosine will now initiate signaling cascade. So let's talk about the different adapter molecules. And we already mentioned the first one, RAS. So this is just a review of your slide. We mentioned the previous one. We're going to go a little bit deeper what RAS does, but we already mentioned it. So remember is that the RAS protein is small GTP binding protein. And remember it serves as a switch key for the control of growth and differentiation. We mentioned what's going to happen when it doesn't work, when there's a problem. People might develop neurofibromatosis. Okay, so this is what it does. So what's the problem with RAS? RAS doesn't contain SH2 domain. So what does that tell you? RAS cannot attach directly to phosphorylated tyrosine. It needs to have an adapter protein that has that has SH2 domain that can attach to it. So what does the RAS do? RAS binds to adapter proteins that contain SH2 domain that attach to the phosphorylated tyrosine residues. So now RAS and adapter proteins will trigger, now we talked about it, series of serine-threonine phosphorylations known as MAP kinase pathway. You remember? They will activate MAP kinase, 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 MAP kinase, kinase, MAP kinase, and all the way to the cell. So this is important to mention. So we have adapter protein binding to phosphorylated tyrosine. Then, because it has SH2 domain, now you have RAS attaching to that adapter protein because it doesn't have an SH2 domain, and it gets activated and stimulates MAP kinase pathway. So that is the importance of this process. And again, we talked about it in previous lecture. Let's talk about another adapter molecule known as STATs. So this is actually known, this STAT means a signal, transducers, and activators of transcription, STATs. And how do they work? They actually contain, look, their SH2 domain. So what does that mean? These guys can directly bind to phosphorylated tyrosine. They don't need another adapter protein. Okay? That is their characteristic. So signaling cascade is the ligand binding will cause receptor dimerization and tyrosine phosphorylation. Okay? Through intrinsic tyrosine kinase in the receptor. Now phosphorylated tyrosine will now attract STATs. STATs can attach to phosphorylated tyrosine because they have SH2 domain. They have that receptor for it, okay? So when they bind to this, is now the receptor will, tyrosine kinase will now actually phosphorylate STAT. So STAT will attach to the phosphorylated tyrosine, okay? And now it's going to get phosphorylated too. So now this phosphorylated STAT will bind to another STAT. So it will dimerize. There's going to be two STATs together. And now dimers will travel to the nucleus. They will bind to DNA, and they will regulate the expression of certain genes. See how deep that pathway goes. So this is not arrest. These are STATs. They have a different pathway. They have SH2 domain. They can bind directly to the phosphorylated tyrosine on receptors. They get phosphorylated. When they get phosphorylated, they then bind to each other as two dimers travel into the nucleus. That is a STAT signaling. I'm going to mention another adaptor proteins known as PI3 kinase. They're very important in promotion of cell survival and cell growth. And how do they work? I'm going to show you that pathway. I'm telling you all these pathways because they're going to be mentioned. They're going to be mentioned in pharmacology. They're going to be mentioned further in physio. So we'll hear all those pathways further down. And I'll tell you, you as a physician, practicing now in the 21st century, we are now making drugs that affect these pathways. We're not anymore just killing bacteria with just popping them some antibiotics. What we want is we actually start affecting the entire signaling cascade. That's the key for treating cancer and everything else serious. So that's why these are important. So this is another adaptor protein known as PI3 kinase. So it's very important in promoting cell survival and cell growth. It stimulates cells to survive and grow, okay? So what happens? You have ligand binding to catalytic receptors, which will initiate receptor dimerization, two receptors attached to each other. Now we know there's going to be activation of the tyrosine phosphatase inside that will now phosphorylate tyrosine in it. The receptor lights up, okay, gets active, okay? Now, PI3 kinase will bind to these phosphorylated tyrosine residues. It can bind to them directly. Now, activated PI3 kinase will phosphorylate a membrane molecules known inositol phospholipids on their third position. So they will actually phosphorylate phosphatidyl inositol 4,5-biphosphate. You remember this guy? PIP2. Okay. So PIP2 is now converted to inositol 3-phosphate, or PIP3, by the action of PI3 kinase activity. We see here. We see the phosphorylated tyrosine. We see the attachment of PI3 to it. And now it will work on PIP2, phosphatidyl inositol biphosphate, that will be converted in phosphatidyl inositol 3-phosphate, which will now be a signaling molecule for this process. Now, phosphatidyl inositol 3-phosphate is now a docking site where it's active for other intracellular signaling proteins known as ACTs. Okay? They will act on it. Okay? So ACTs will bind to this phosphatidyl inositol 3-phosphate, or PIP3. Now, ACTs are also known as protein kinase B molecules, and they're recruited by phosphatidyl 3-phosphate. And what the ACTs are going to do, I'll tell you why it's all bad. Interesting. In bad. That's a good one. So, is because ACTs will now phosphorylate BAD, another molecule. And phosphorylation of BAD will result in its inactivation. So this whole process makes BAD disappear. Okay? You don't want BAD in a cell, do you? It's bad. Okay? And I'll tell you why it is bad. Now we go back on a big picture. If BAD is not inactivated, if it is active, it's a bad thought that pushes cell to death. BAD is a signal that will tell cell, pull a gun and blow your head off. Okay? So if BAD is active, cells die. They choose to die, they go under apoptosis. So what does this signaling mechanism do? Inhibit BAD. It makes your cell survive. Your cells choose to survive because of this entire pathway we talked about. PIP3, PI3 activates PIP2 into PIP3, and then what's going to happen is it will recruit ACT, ACT will phosphorylate and inhibit BAD. Whole process. It's very important. Now this process results in cell survival, but this process can be terminated. What's going to happen if the cell is funky looking and chooses to die? You know, there's some problem with the cell. So your body wants to get rid of it. The best way to get rid of the cell is, I don't want to kill you, you kill yourself. Okay? So that is the system. That's the system for cells. So what happens is, there's going to be termination. PIP3 will be broken down by the enzyme known as PTEN. So PTEN will break down PIP3, and BAD will become activated, and the cell will kill itself. Okay? Makes sense? Okay? So that is, PTEN is going to be breaking down and inhibiting the entire pathway. So what's going to happen if you have a mutation of PTEN? You have a cell that is funky looking, and it smartly wants to become a cancer. That's not true, but I'm kind of thinking you can best think about it. I want to become a cancer. So what the cell does, it has a mutation on PTEN, makes abnormal PTEN. So now cell is abnormal, and it should receive a signal to die. But it doesn't. Why? Because PTEN doesn't degrade phosphainositol 3-phosphate, and BAD is constantly inhibited. Makes sense? So the BAD is constantly inhibited. It cannot kill that cell. So cell survives. Okay? So the cell will produce abnormal PTEN enzyme, which will now not break PIP3. PIP3 will still cause BAD not to work. So the cell will survive. That's a serious problem. And we found that mutations of PTEN, as found in glioblastoma multiforme, tumors of the brain, we find that are found in endometrial cancer and prostate cancer. So these cells will prevent their own suicide, okay, by mutating PTEN. Now let's talk about signaling through non-receptor protein kinase. So what is this? Non-receptor protein kinase are actually, remember, we talked about receptors that have their own protein kinase in there that will phosphorylate tyrosine. Remember, we mentioned those. Now, we have receptors that do not have that. They have to recruit, in order to activate themselves, the receptors, they need to recruit the protein kinase inside the cell, some other protein kinase, not their own, not the one that they have. They don't have any in their chain. So those are the receptors. So some receptors, specifically for cytokines, very important in your immuno, for interleukins, interferons, and some hormones, prolactin and growth hormone, do not possess their own tyrosine kinase activity. So what they need to do, they need to activate other tyrosine kinase inside the cell to phosphorylate the tyrosine and light up. So that is the process. So cytoplasmic domains of these receptors will form non-covalent bond. So you know those tails? They will bind to these tyrosine kinase from the cell. They will recruit the tyrosine kinase, and tyrosine kinase will now phosphorylate their tyrosine. Okay? So they will not have tyrosine kinase in them. So then that will result in the activation of the receptor. I want to mention two tyrosine kinases that can be recruited by the catalytic receptors, and these are this is SRC and Janus kinase. So SRC protein kinase, there are at least 8 members of this family, of those SRC non-receptor tyrosine kinase family. Protein kinase, but specifically tyrosine kinase family. So this is how they look like. They have 3 domains. They have SH3, SH2, and SH1. Now, if you remember what I mentioned to you, what SH2 will be used for? To attach and bind to the receptor. Doesn't have to be phosphorylated. They will phosphorylate it, but need to attach to it, okay? SH1 is actual enzyme. It's actual kinase. It's actual tyrosine kinase. And there are different members of this family that are found in different cells. And we also know that various members of the SRC family can phosphorylate tyrosine residues in many of the same target proteins. So what does that mean? Several SRCs can work in the same protein. You know, so there can be different members. It's really complex. Now, SRC family members are actually regulated by phosphorylation of tyrosine residues on them and by protein and protein interaction. So SRC proteins have to be are normally inactive, and they have to be switched on only at crucial times. You know, these SRCs, you don't want them to phosphorylate tyrosine all the time. They want to phosphorylate tyrosine when the signaling is happening, okay? So they are all normally off, and they will be activating when they're needed. So clinical significance. If SRCs remain active, you're going to end up with cells that will grow abnormally and will become malignant. So mutation in SRCs are actually found in multiple cancers. The next group, and SRCs are boring, okay? Let's hit the cool ones. You know, I know you like now, at this point, it's a masochism, you enjoy it. So, you know, we're going to go through another pathway. It's kind of fun. So this is another external kinases. These are known as JAKs, or also known as Janus kinases. So this is it. You have a binding of the ligand to a receptor that doesn't have its own tyrosine kinase, okay? So it binds to it, causing dimerization. The next, this will result in attachment of JAK, okay? And the JAK will now phosphorylate tyrosine on this cell, on this receptor, lightening up the receptor, or turning on the signaling, okay? So again, JAK will attach to it and cause phosphorylation. Now, we're not done yet. The next molecules that will now attach to these phosphorylated structures are STATs. We talked about STATs already. So now we have STATs attaching on phosphorylated tyrosine amino acids on this receptor. So first JAK phosphorylates them, then STATs has SH2 domain and attaches to phosphorylated intracellular portions of the receptor. Now remember, the STATs get phosphorylated. We mentioned this. And now we have STATs phosphorylated. Two STATs will bind to each other. And what they're going to do, they're going to travel all the way into the nucleus, where they're going to affect the cell transcription, DNA transcription, and everything else. So that is the process of this. This signaling cascade is known as JAK-STAT pathway. Okay? So this is a JAK-STAT pathway. So you remember, JAK needs to phosphorylate tyrosine in order for STAT to bind. STAT gets phosphorylated, finds its brother, joins it, and travels all the way to the nucleus where it regulates the DNA transcription. Now, why is this process important? It affects cell growth, it affects cell differentiation, affects cell death. This is a very important signal maintaining a normal survival of the cells and regulation of cell survival. So disrupted and dysregulated JAK-STAT signaling will result in immune deficiency syndromes and in cancers. And you're going to hear that. So now when you hear somebody discuss in immunology the condition, they're going to say there is a problem with JAK-STAT pathway. You know what it is. Janus kinase. At least you're going to remember that. You're going to remember that is the no, you're not going to remember that, you know, in the year. But, you know, you're going to remember, oh, Jack. Yeah, it's like, oh, I know Jack, his brother Bill. But the thing is, you know, the JAK was, you'll remember this Janus kinase. It's receptor, hopefully remember, that actually doesn't have its own protein kinase or phosphorylase. Tyrosine kinase. So what's going to happen is this cell needs to recruit JAK. JAK then phosphorylates and activates STAT. Now, why is all this important? Is because this is important to me. You know, I am a type 2 diabetic for a while. Everybody in my family has it. So just I'm going to touch upon of insulin signaling. This is a little sidetrack. So now you're going to learn how insulin works. Just to tell you guys what insulin does. Very, very generalized. I'm not going in that. You're going to hear that all over in physio and pathophysics. But this is it. When you eat, of course, you get a lot of carbohydrates in your blood. You know, you eat bread. Everything has carbohydrates. Fruits, vegetables, everything has carbohydrates. These carbohydrates go through your blood. They will now, you need to take these carbohydrates into your cell to produce what? To produce energy. To produce ATP, you know, because carbohydrates are a really good source of energy. So whenever you eat, your carbohydrates level in your blood go up, okay? But you need to push these carbohydrates into the cells. You need to get them there so they will be used for energy. That's what insulin does. Insulin stimulates signal cells to take up those carbohydrates and use them for energy. Okay? So individuals who have diabetes have that problem. I eat cake and my blood sugar lasts forever. Okay, it's high. For a normal person, it will be in an hour down. For me, in an hour, 2 hours, 3 hours, my glucose levels will be still high. It's not why, because my insulin signaling is not working. So this is insulin signaling. And now you learn it, you say, aha. Maybe you will. If you don't, just say aha, okay? But this is what it is. It's actually insulin signaling is through this catalytic receptor. That's an insulin receptor, okay? So insulin receptor is a catalytic receptor that contains its own tyrosine kinase. So think about it. It doesn't need Janus, JAK-STATs. It doesn't need all that. Blah. It has its own tyrosine kinase. Interestingly, that insulin receptor is actually already dimerized. So it doesn't need the ligand to dimerize it. It's already 2 chains next to each other, joined together. So binding of insulin to extracellular binding domain of this receptor will result in activation of tyrosine kinase in it. Tyrosine kinase, you remember, is going to phosphorylate those tyrosine in the cell, lighting it up. Now, this will result in the activation of various insulin receptor intracellular substrates, known as IRS proteins, internal revenue service proteins. Okay, so why are they why you're going to remember them as internal revenue service? Simply because they will go everywhere, and they will. So, you get them on your back, they'll find you. So this is what it is. So these IRS substrates are several. So think about it. You have one signal of insulin, and then it breaks down to several IRSs, several signals all over the cell. So it will stimulate IRS1 and IRS2 pathways, IRS3 and IRS4, insulin receptor intracellular substrate, and will cause different things through the cell. So one single signal can cause tons of signals to pop out and work on different spots. It's really, really extensive signal. So phosphorylated IRS will promote activity of various protein kinases, phosphatases, and will result in these multiple effects. And this phenomenon is known as signal splitting. One signal that will split to several signals in a cell. That is the characteristic of insulin signal. So what's an insulin resistance? Some individuals have type 2 diabetes, individuals who have metabolic syndrome, and I'm going to hear from Dr. D, metabolic syndrome, I'm actually teaching it next week for second years, is a condition with individuals who have certain circumference of their abdomen, have high triglycerides, have low HDL. There is a whole set of things that have insulin resistance are considered to have metabolic syndrome. Metabolic syndrome is usually a precursor for diabetes. The patients who have, or cardiovascular disease and everything else. You have patients with metabolic syndrome. You as a physician, half of you will have metabolic syndrome. Why? Because of our lifestyle, because of the genetic things we carry, we'll end up with metabolic syndrome. The question is, will you let it take you? Or are you going to hit the gym once every 6 months, like I do? Or, what you do is, as a doctor, tell your patients to pay gym membership, and that's enough. You don't have to go to the gym, because that helps you prevent diabetes. Okay, so you just go there and say, how much not to have diabetes? So 500, here you are. Yeah, but that's what it is. So that's a metabolic syndrome. But let's go back, not to digress, there are conditions where actually insulin receptors don't work well. This is in metabolic syndrome. This is known as insulin resistance. What does that mean? Is your body produces insulin, but receptors don't work well. Signaling pathways are messed up. That's why we don't know cure for diabetes yet. Because there's all things can go wrong. So what happens with most of the people with type 2 diabetes like mine, my insulin levels are high. Type 1 diabetes patients don't have insulin. They don't produce insulin. You're going to hear about it. I'm not testing you on this. But the type 2 diabetes patients have high insulin. My parents have diabetes, my grandma, everybody, genetic thing in my family, type 2 is shown to be genetic, type 1 not. What happens is that type 1 is connected with you getting some infection when you were a kid or some autoimmune response that destroys your beta cells. I'm going all the way down. Let's talk about insulin. Forget this lecture. So this is it, you know. But what it is, you know, so one of the things is actually that there is a problem. So, for example, I have high levels of insulin. So since I have high levels of insulin, that's not good for you. Previously, we thought, oh, you know, type 2 diabetes, give them more insulin. Oh, great idea. I already have high, so give me even more. So it will overload my, you know, receptors, so it will work. So I start pumping myself with more and more and more insulin. We now show that insulin is bad for you. So you're actually doing worse. So those are the things that will happen. Guys, find a cure for diabetes. You're going to get flowers from me every week and a lot of fame, okay? But that's what it is. So again, insulin signaling affected by different individuals. Let's talk about steroid receptor signaling. So steroid receptor signaling is another one. That is dear to me. Why? Because I worked on this thing during my PhD. Steroid receptor signaling actually work with retinoic acid and vitamin A and their effect on heart development and kind of weird stuff. But what it is that now we have intracellular receptors for steroids. This steroid receptor signaling, what are the steroids? These are hormones, okay? And these are the hormones who will signal differently. If we talked about some hormones attaching where? To the receptor, activating the whole, you know, catalytic pathway, everything else. Steroid hormones don't need that. They're fatty. Steroid means that they're made from steroids. So what does that mean? Is they're actually made from cholesterol, okay? So these hormones do not need to attach to receptor. They will go into your cell, will penetrate the membrane, and attach to receptors inside the cell. So they will have receptors inside the cell, deep inside, not on the surface. So those are steroid receptors. So the intracellular receptors for steroids are located inside the cytoplasm or inside the nucleus. So what is it? These receptors are interesting. Why? Because they're ligand-activated transcription factors. When you attach steroids to this receptor will now regulate your DNA transcription. So it's very, very powerful thing. So ligand and hormone will cross the membrane, will bind and activate to intracellular receptor proteins. Now, receptors will also most commonly found all the way into the nucleus. Receptor activation will enable them to bind to DNA, and after binding to DNA, these receptors will now regulate transcription. So what they're going to do is they're going to regulate the transcription of different genes. Now, this gene, of course, is translated into protein, and the classical form of steroid signaling is known as nuclear-initiated steroid signaling, or NISS, which is the signaling through the nucleus, all the way into the nucleus. Using this pathway, the steroid hormones will have direct effect of cell genome. So why these steroid hormones are important? Why do they affect your genome? Steroid hormones are testosterone, estrogen. It changed us. Remember teenage years. Nobody wants to remember that time. We start growing hair in weird places, okay? We start feeling confused. So that is it. That is all steroid signaling. It actually can affect completely a DNA. It's very, very powerful. That's why I love them. They're really awesome. And these are not only, reproductive hormones. There are a lot of other things. So important note is this, guys, it takes longer time. They're not that fast. You know, you did not wake up in the morning and said, oh, I need to shave. Which is awesome, overnight. You know, you remember that old movie from my times, Teen Werewolf? He wakes up like, he's already here all over. No, it doesn't happen then. It takes time. So it's a slow process, but it's very powerful. It can take minutes, hours, days for the effects to work, but it actually are very important because it takes time to produce that protein. You learn this. Protein doesn't happen boom, boom, boom. Expression of gene doesn't happen overnight. Otherwise, we will be Spider-Mans. We'll be bitten by that, spider and become overnight a Spider-Man. No, even the Spider-Man movie took some time. We're going to geek phase now. But, so what it is, it takes time, okay? So that's the characteristic of these. So, now let's look at it. In order to bind and activate the intranuclear receptors, steroid hormones have to travel. These guys are travelers, okay? So they need to go all the way from circulation through the cellular membrane, through the good old cytosol, all the way to nucleus, where they bind and regulate DNA expression. But remember that these steroid hormones are all made from cholesterol. That's why cholesterol is not that bad for you. Oh, wait. So that's a precursor. They're cholesterol-made they're steroid compound. So they're synthesized in adrenal cortex, where you're going to produce cortisol. You're going to hear about it. That's the stress hormone. Aldosterone, that is a hormone regulates water activity. Again, I'm not going to test you on it. I'm just telling you what they do. And androgens, of course, the hormones of male reproductive, precursor for male reproductive system, male reproductive hormones. Then ovaries and placenta will produce estrogens and progesterone, and testes will produce testosterone. They have this specific structure which enables them to cross the membrane. Why? Because they're fat, so they easily go across the membrane. And these hormones play a very important role. Cortisol in stress. Sexual function is testosterone, estrogen, eventually progesterone. Also, aldosterone will regulate the activity of kidneys. Kidneys, it will regulate how much fluid do you produce by the kidneys or secrete. So how do they work? Is estrogens are transported for their original site of production into the blood. So wherever they're produced, they will be sent in the blood. Okay? Now, in the blood, why? What's the blood? Is the blood fatty or watery? Watery. And we are talking about what? We're talking about fatty substance coming from cholesterol. So those hormones, steroid hormones, will not like to be in blood. So what do? They find a powerful protector to carry them through the blood. So they will bind to a carrier protein as they're carried through the blood. Mainly, it's albumin. A large protein in your blood is going to carry them, protect them from water. There are other specific carrier proteins, but albumin is the most important. So when they reach the cells where they need to work, they will be detached from albumin and they will cross the membrane due to their lipid nature. Inside the cytoplasm and nucleus, they will encounter intranuclear or intracytoplasmic receptors, and hormone binding will modify this receptor, and now this receptor and a hormone together will now affect the activity of the genes. So let's talk about breast cancer. So we're going into clinical situation. So we know that certain breast cancer cells are known to have large amount of estrogen receptors. So these breast cancers have huge amount of receptors for estrogen on the surface. Why? Breast cancer needs some, not all breast cancers, but the group of breast cancers, specific group called estrogen receptive breast cancers, they have large amount of estrogen receptors on their surface. Need estrogen to survive. They need estrogen to actually grow, stimulate their abnormal growth. Okay. So this is a characteristic. So we see that this signaling will be used by cancer cells in order to promote their growth. Now there are types. These estrogen receptive breast cancers are actually very commonly found in postmenopausal women. Okay, specifically in postmenopausal women. But there is a problem. And I'm going to explain you with the difference between estrogen production and why there is a different types of cancer affecting women postmenopausal and premenopausal. Okay? Postmenopausal women have had their last period. Their production in their ovaries of the steroid hormones goes down. But of course, they don't stop being women. So what do they do at this situation is that postmenopausal women, the main source of estrogen are peripheral tissues and various regions of the brain. What the women will do? They will produce estrogen from fat cells. They will actually produce estrogen from brain. And they will actually steal, or we all have some androgens, male hormones in ourselves. So women will use those androgens now to produce estrogen. Okay? So that's the postmenopausal women. Premenopausal women, we know, produce estrogen in their ovaries. So there is going to be a difference that you're going to see that relates to the therapy of cancer. So in postmenopausal females, the main source of estrogen is actually aromatization of adrenal-produced androgens. So what does that mean? Is the main source is process of utilizing the male, in a sense male, precursor of the male hormones. They will actually use those and convert those androgens into estrogens. And that is achieved by the action of the enzyme known as aromatase. Okay? That will actually cause this. So how can you treat women, postmenopausal women, treat the estrogen-sensitive breast cancers? By decreasing estrogen. How will you decrease estrogen in postmenopausal women? Their ovaries don't work. So what you're going to do is you're going to affect that conversion. So you will give aromatase inhibitor drugs. So we'll inhibit that conversion and production of estrogen. Okay? So less estrogen, less cancer cells will grow because they need estrogen. So that's one of the therapies that is actually given. Now when the cells, the breast cells, cancer cells don't receive enough estrogen, they'll choose to commit suicide, okay? Or they will be destroyed, they will not grow properly, at least you can control their growth. So post-menopausal women, you will give the aromatase inhibitor drugs. This is all example of this steroid pathway signaling. So again, I'm just going to touch upon again, I just want to review this, a type 1 steroid signaling process. So we have a hormone that will travel to the cytoplasm and nucleus, where it's going to bind to a hormone receptor. Now this will result in the activation of the receptor. An activated receptor ligand complex will associate with another molecule known as co-regulator or co-activator proteins. So you're going to have what? You're going to have, for example, a steroid hormone will attach to its receptor, and it will attach to co-regulator. So all these three will play a very important role. Now receptor ligand co-regulator complex will now bind to regulatory DNA sequence that regulates the DNA transcription, regulates the normal function of the genes. So it will bind to HRE segment through the zinc finger motives. That's a specific type of attachment. Did you already heard about zinc finger motives? You're going to talk about it probably very soon in your lectures when they talk about, Dr. Kuo might mention them. That's the way how they attach to the DNA, these complex. When you have steroid hormone, you have receptor, and you have co-regulator. They will go all the way down to the DNA, and they will attach to this region of the DNA that is known as ARCH-RA with these zinc finger-like motives. It's going to dig in and stop there. This binding will enable now this regulatory domain on the receptor to interact with proteins of transcriptional complex bound to gene promoter, and they will stimulate the activity of the genes. So what do? They bind to HRA, okay? And now this co-activator that we mentioned will now promote certain genes, will stimulate the transcription of specific genes. Now, last but not the least, let's talk about how do we treat breast cancer in premenopausal women. We use, what is the problem with premenopausal women? Now you can't block, there's a big problem with premenopausal women. Somebody smiled to me, I'm just joking. But there's a problem with us, let's put it that way. I've been married too long, guys, to know that everything is my fault. Okay, so weather is a problem, war in Syria, definitely my problem. All of you guys in long relationship married, that's the key for successful marriage. Walk in, it's my fault, it's fine. Yeah, did you hit your car, honey? Yeah, my fault. It's like, yeah, it's fine. Yeah, it's because I made you drive that car, which is better than my car, but that's okay. So, yeah, what would we do without each other? Nothing, My wife would have anybody to blame. So, and Cat, she loves the cat more than me, so Cat will never be blamed. So it's me, Cat has diarrhea, your problem. Why? You fed him food. So he asked for food, for God's sake. It's a cat. But, and then she's like, never feed him. Now I'm going totally different. Never feed him human food. And then she's like, kitty, 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 you're feeding him a human food. It's like, no, you do it. I don't know, guys, if you start doing diet, and of course that happens in my house very often, for both of us. Mine lasts 30 hours, my wife's lasts a little bit longer. At least she says that it is. But what it is like, and then I buy all these things, treats, and I hide them somewhere. I'm diabetic, I shouldn't be eating those stuff either way. And, and my wife finds it somewhere hidden, and she says, you did it. You bought it again. I was like, why were you looking for it, for God's sake? I'm running out of ideas where to hide it. I really am. It's like, and once I bought stuff, I remember in the store, and tons of candy and stuff which I shouldn't eat. Again, that was after exam, one of the med school exams. Trust me, we go through motions like you do. So I'm like having all these candy with me, like they did well. I'm celebrating, okay? So I'm like, and I'm buying. Lady says, like, what you gonna do? I said, I have to hide it from my wife. She says, you know what you should do? Yes, hide it in your car. That's what my husband does. I was like, awesome. So guys, if you need anything, just go buy my car. I have some candy somewhere. It's like, it's somewhere hidden, you know. You find it 3 months later, who cares? Candy is a candy. But, you know, that's the idea. So let's go back to this. So, premenopausal women, they, of course, you can't affect their aromatase. Why? Because they're produced, and they need, another thing is they actually need estrogen. Why? Because they actually need to have a normal effect of estrogen on their bodies. You don't really want to completely shut down their estrogen. Okay, so what you do is you use selective estrogen receptor modulators, such as tamoxifen. You probably heard about it. It's the drug when it came out, it was a revolution, decreasing the cancer incidence. And everybody all over CNN, everybody tells it's the best drug ever, but you don't understand, not all breast cancers are estrogen receptive. So yes, there is a population where it works, but not everywhere. But this is it. So these selective estrogen receptor modulators, what do? They bind to the estrogen receptors on the breast cancer cells. So when they bind, they inhibit the binding of estrogen. So they kind of lodge themselves on the receptors, and they do not allow binding of the estrogen. So these cells don't receive it. They don't receive the stimulant as an estrogen, and actually cancer dies. So not dies, but it definitely decreases in size. But there is one problem. This is not in your board, but I just want you to know, because now, guys, you're doctors. People will ask you, advices. So you will look at moles and different growths on people's bodies. You say, we need to remove that or not. So one of the things is this, now, tamoxifen has another problem. Although it inhibits the breast cancer receptors, it stimulates endometrial receptors. So these women have higher risk of developing endometrial cancer. So you kind of have to follow that. You have to be very careful because, again, drugs are never perfect. There's a negative and a positive effect of the drug. Okay, guys? I think that's it. Thank you for your attention. I'll see you tomorrow. Have a little break and enjoy whoever comes next after me. Who's coming? Oh, Dr. Kuo. Awesome. Thank you very much, guys.